Dear Chairman, dear audience, first of all, thank you for this great opportunity to present our research area here in this nice event. And as you can see, I chose a bit old title, but during my talk you will see that it is related to the model organism what we are using to study innate immunity, because actually this model organism is a soil sentinel. So first of all, I would like to talk to you a little bit background of the immunity, immune response. As for humans, to defend our host body, it maintained at 3 levels. So we have so-called more passive anatomical physiological barriers, such as our skin building epithelial cells, or the gut building epithelial cells also in the airways. But if this more passive anatomical physiological barrier is injured, then comes in action the second level of our host defense, the innate immunity and inflammation, which provide a very fast, prompt immune response against pathogens. But very often this is not sufficient, because many pathogens already learned how to trick out the innate immune mechanism. So then comes our third level of host defense, adaptive immunity, the highly specific, so-called antigen-specific adaptive immunity, and both mechanisms with cellular and humoral components fight against the pathogens. So this is true for humans, but not just for humans. From fishes, actually from shark to human, we can observe innate immunity and adaptive immunity. And I brought this image, this jigsaw puzzle, just to show this is more like an artificial distinction. In reality, to stay as healthy, these 3 levels need to actively interact with each other. But I told you that adaptive immunity is present only in vertebrates, but there are many other organisms, actually invertebrates. And invertebrates, they do not possess adaptive immunity, but they have a very sufficient innate immunity and inflammation. So I will talk to you about a very unique invertebrate model, which actually not really widely used in immunity studies, because if we hear the term immunity, then we most often think about human and mice studies, maybe Drosophila or C. elegans, but we are using a totally different model organism. We use segmented worms, which they belong to the annelid phyla. We use earthworms. There are many unique anatomical and physiological qualities of these animals, because they have a closed circulatory system, and also their blood contains hemoglobin, which transfer the oxygen. And they have also a real body cavity, so-called colon, and the immune function is related to this colon because it's filled up with a fluid, and this fluid contains many different molecules, namely antimicrobial molecules. Here I listed the well-known lysine. Yes, some have also lysine, not just we have. And also other unique molecules, such as the lumbricin antimicrobial factor. And in addition to the humoral components, the coelomic fluid also contains cellular immune components, so-called coelomocytes, derived from this coelomic name. They have different groups of coelomocytes participating in the immune function. One group has this highly granular cytoplasm, so-called allocytes. You can see better with this transmission electron microscopic image. The other group are the so-called amoebocytes, and typically they are attached to surface. They building philopodia. You can also see better this scanning microscopic image, both. Okay, what is their function? Actually, amoebocytes are able to phagocytose, to engulf pathogens very actively. Here is just an example when Staphylococcus aureus bacteria was phagocytosed. But these cells can, and the animal can encounter not just microbes, but larger pathogens. And we modeled actually that the coelomocytes are able to isolate, so-called encapsulate, these larger pathogens from the environment. This is the so-called encapsulation. But I made a mistake. I should start actually my talk that how I came to work with this unique species, unique group of animals. Actually, I was interested in immunology, and I knocked door on the Department of Immunology and Biotechnology in Pécs University of Pécs, and then the department head, Professor Német, welcomed me. And in the first more than a half year, I was just only doing lab work, learning different laboratory methods like cell culture, cell and tissue culture, immunohistology, immunoserology. And after a time, after this half year passed, I was quite already anxious to have a final thesis topic. So I again knocked in his door to, okay, I'm here and I would like to have finally a thesis topic. And he said, okay, finally here is a biology student, so I can work with him, giving him this earthworm model. And to tell you the truth, I was quite puzzled because at that time everybody in the lab worked with mice and maybe some human studies. Nobody worked on worms. So he said me, okay, let's start to collect literature and try to start the research. And I take this challenge. I found 13 articles, discussed with him, and then started to do the first just a thesis work and then a PhD. And since PhD, I'm still working on this animal with different aspects. So in addition to immunity, actually these animals has a unique feature that they can very easily regenerate their segments. And our current interest turned to regeneration because if you see this phylogenetic tree, I try to emphasize that different organisms have different regeneration capacity. Actually, cnidarians, so jellyfish, are able to regenerate their whole body. Also flatworms. So, for instance, if we put a small cnidarian, a hydra, into a Eppendorf tube, we homogenize the hydra. In a couple of days later, they again rebuild their body. So the individual cells, they know their identity, and they rebuild their whole body. Also, a very unique regeneration, as I told you, it's true for flatworms. If you cut these worms into 3 parts, all the 3 will regenerate the head, the tail part, and they will be again a full individual worm. However, if in evolution we are getting a bit further, in segmented worms, so our models in annelids, we don't find whole body regeneration, only body part regeneration. This is also true for certain arthropods, or even in insects, we can only find certain tissue or cell regeneration capacities. And this is also true for vertebrates, that in fish, they are able to regenerate their fin and also intricate internal organs, for instance, eye, in the eye, the retina, or even cardiac muscle. Also, certain amphibians are able to regenerate intricate structures, or reptiles. I think you all know that lizards are able to regenerate their tail, but it's less known that amphibians are also able to regenerate their limbs. However, mammals, we have a much lower regeneration capacity. We are able to regenerate actually certain part of our body. It is known well that the liver is able to regenerate for a certain level. But why this regeneration capacity is going restricted upon evolution, we don't really know. But there is an interesting observation as the immune system is getting more and more complex, then the regeneration capacity is actually oppositely correlated. So we started to observe the regeneration in annelids, in earthworms, and here I just show you the anterior part, so the head part. So when we injured the head part, upon 2 weeks, a small transparent tissue, so-called blastema, grown back. And after 4 weeks, we were able to see already the regrown segments. This was also true for the posterior part. Here, with this GIF image, you can see better how this is changing. So just here, when the intact is this first cut, then the blastema is growing, and the new segments are appearing. This is for the tail part. This is the intact. Then the first regeneration blastema starts, and after 2-3 weeks, you can see the regrown segments. This data was mostly done with one of my colleagues, Cornelia, and we were also interested in not just the morphological features, but also how the actually the cells are proliferating during regeneration. And for this, we applied a trick. We injected with a special chemical the regenerating worms, and after we cut the regenerating segments and used a reagent to have these beautiful images to observe in the dividing cells. This is the regenerating blastema, and this is the intact segments for the anterior, and this is for the posterior region. Again, this is the blastema, so the newly grown segments, and these are the intact segments. And you can see where the green dots, those are dividing cells. And I just put here this picture because Cornelia was quite upset when I told her that, okay, this is a nice thing that we observe with microscopy, but let's quantify it. So I asked her how about to count it? So she was not very happy about this idea, but finally she managed. And after this, on one of her presentations, she put my face onto the image. But yes, we managed this. And as a follow-up study, actually, we were interested more that what type of molecular backgrounds could be happen during regeneration, and we chose a well-conserved, well-known developmental morphogenic pathway, the so-called VNT beta-catenin pathway, which is known, the different components of the VNT beta-catenin pathway is known from Knillerians, but actually in earthworms it's not really identified yet. So the central component, the so-called beta-catenin molecule, we were able to characterize it and describe the whole sequence. And after the molecular characterization, we were also interested whether it is participate in the regeneration. So again, we did segments, sections, and stain for beta-catenin, also with this serpoproliferation dye, and we were able to detect that actually they were sort of reversely interacting during the first phase in the 2 weeks of the regeneration. And in this case, I tell you, Cornelia was a bit more happy because in this case she didn't have to count it by microscopy, because the microscope we used a HTS, a special microscope which counted the software counted itself the positive cells. So she was less tiring. I gave her a less tiring job. And I just want to show you that this is another follow-up experiment was where we exposed worms to different pathogens, different bacteria, yeast particles, and also mimicking viral nucleic acids. And after different time points, at early time points, we already observed that bacteria, pathogen incubation, and also in later time points, they induce the beta-catenin expression. So to sum up, I just would like to give you some take-home message, not really related to this research area, just generally, that what I can recommend you to not just find an interesting topic, but try to find such a PI, so a research leader who is supporting you, because I was lucky for that. And also I suggest that science is quite interesting, so that's also should be a boost that we do science because we are interested in, and we ask questions and we try to find answers. The key is to find the right questions and find the right answers for it. And I didn't tell you more details about it, but research is also sometimes can be not always on the good side, because to tell you the truth, in experiments many things can go wrong. And in, let's say, 20% of the experiments can be good, but on the 80% can be going wrong, and then you need to figure out what's going wrong. And that's also a challenging thing. So I recommend you don't give up, just try, think it over, what you made wrong. And first, look it in your studies, that might be something was wrong with the recipes or the project. Okay, so, and also, I want to say that this is not a one-man show. Of course, many people helped me to work on this project. First of all, the department head, the research department had Professor Berki, and I mentioned to you Péter Engelmann. He was an active fisherman, and actually that's why he came to this idea that when he was doing the angling, then, okay, he had an injury on the finger and got a very serious inflammation. And he had the idea, okay, these earthworms are living in the soil, they can deal with this whole bacteria and microbe environment quite well, so they should do something well with their immune response. And I also would like to thank my colleagues, my PhD students, Cornelia Bodo, I mentioned already her name, Bogdana, they both worked not just on regeneration, but Cornelia mostly on regeneration, but also nanoparticle uptake, also Bogdana. And recently with Cheyenne, we are starting a work to more studying the epigenetic part of the regeneration. And of course, the dirty deeds that science needs some funding. So these are the funding that I received from the university to maintain this research. And you also need a solid background from the family, so the knowledge now comes to them. So unfortunately, last year I had 2 serious losses. I want to tell that my parents gave very big support during all those studies. Unfortunately, my father passed last year, so he cannot part of this honor now. And I also would like to tell that my partner, so my wife, gave a very large support. Unfortunately, she can also not be here. But also my children, because they also take, they can share this honor, they gave a lot of activities, a lot of energy, but they also take out a lot of energy. And finally, I would like to thank you for your attention. I hope I didn't take too much time of you. And I would like to thank your attendance and attention with this funny cartoon. This is a satiric cartoon from the Punch Almanac, which actually shows Darwin emerging from chaos through earthworm to a respective gentleman. And many of you know Darwin from his work, Origin of Species, but not so many of you are known that actually Darwin worked on earthworms. His first publication for the Geological Society was on earthworm 22 years before he published the Origin of Species. And 20 years later, as he published the Original Species, his last publication, a book, was about mold information by earthworm. Thank you for your attention.
Thank you for the presentation. Are there any questions? If no, I would like to ask you for a group photo.